Hello, this is Avril Dilemma, and I will be talking about DNA methylation using this poster that I've made. So, DNA methylation, in simple words, is the transfer of methyl groups to the molecule of DNA, specifically a cytosine base. And in scientific words, we can say that DNA methylation is a heritable epigenetic mark involving the covalent transfer of a methyl group to the C5 position of your cytosine ring of DNA by DNA methyltransferases, DNMTS. Epigenetic, we break it down. Epi is a Greek word which means beyond. So, beyond genetic structural codes, we have external factors that affect your gene expression, and one of them is DNA methylation. So, DNA methylation involves your methyl groups attaching to the cytosine ring of DNA. So this happens in the presence of DNMTS along with SAM, which stands for S-adenosyl methionine. With this is your methyl donor, which brings your methyl group, and DNMTS is the one which attaches it to your cytosine ring. So there are different types of DNA methyltransferases, which I'll get back to in a bit. First, let's talk about DNA methylation and its effect on different regions of a gene. So, when we talk about the promoter region, we know that the promoter region has elements which are essential for the initiation of transcription. It is the recognition site of RNA polymerase and for the initiation of transcription. So, methylation of the promoter region usually represses gene transcription factors like RNA polymerase, making it difficult for transcription machinery to initiate gene transcription. So, what happens when your promoter region is methylated is that it alters the DNA structure in such a way that it attracts methyl binding domain proteins, and these proteins can recruit enzymes that can modify histones, modify the histones in a way that it makes the chromatin more compact and inaccessible for your transcription factors to have an effect on it and bring about transcription. So, which is why your gene is repressed, and when your promoter region is methylated, transcription cannot take place. And then, talking about your gene body, it is the opposite. When your gene body is methylated, it promotes transcription by facilitating elongation of RNA polymerase along the gene, potentially enhancing gene expression levels. In general, methylated at the CpG site, which is a cytosine guanine site. So here what happens is it loosens the chromatin in such a way that it facilitates your transcription when your gene body is methylated. So now here we were talking about the different types of DNA methyltransferases. So the first two that play a role are DNMT3A and DNMT3B. They establish new DNA methylation patterns by adding methyl groups to previously unmethylated DNA. They create a tissue specific methylation pattern. So here I've taken an example of embryonic cell. There is a DNA that we take from it, and it's a wound around your histone proteins. So now this is unmethylated, as you can see. But now we are going to introduce DNMT3 and DNMT3B, which, as I said, are your they establish new DNA methylation patterns, and they create tissue-specific methylation pattern. So this procedure is known as de novo methylation. Novo means new, and methyl groups are being attached to a strand which did not have any methyl groups, right? So, when this happens, your strand is now having your methyl groups, and now it is methylated. So once it is methylated, we talk about another enzymes which whose role is maintenance. That is your DNMT1, also known as the maintenance methyltransferase. DNMT1 is responsible for maintaining DNA methylation patterns during DNA replication. It recognizes hemimethylated DNA with one methylated strand, and adds a methyl group to the newly synthesized strand. So as you can see here, we have the CGP site also, which is where, as I said, methyl groups attach. It's specific. Now, besides your gene where your methyl groups can attach, epigenetic marks like methyl groups don't have to always be on DNA directly. They can also impact the histones. So now, when your methyl groups impact your histones, it is known as histone methylation, which is your histones basically having your methyl groups attached. So this generally tends to prevent transcription. And on the other hand, when acetyl groups are attached to your histone, it brings about histone acetylation, where it loosens the packing of DNA around histones, allowing transcription factors to access DNA. So now this was methylation. Now talking about demethylation, the main enzyme that here comes into picture is TET, and TET is an important enzyme that has a role in regulation of DNA methylation patterns. So here we have 5-methylcytosine, which in the presence of your TET gets converted to 5-hydroxymethylcytosine. Your methyl group from the fifth position of the of your cytosine is removed, and a hydroxyl group is attached. So when this happens, your as you can see, you've lost the methyl group. So after this, it can with the help of some pathways also remove the hydroxyl group and bring it back to cytosine. So this brings about a demethylation. So now, when we have this TT along with your DNMTS, we need to have a proper balance of these in your cell. Otherwise, what happens is when there is an imbalance and the mutations can take place, and specifically cancer. In cancer, you can have hyper methylated strands and even hypomethylated strand, where there's methyl groups present in axis and also not present only on some parts of the gene. All right, and now let's talk about the factors affecting DNA methylation. In humans, DNA methylation is primarily influenced by factors such as genetics, environmental exposures like diet, stress, and toxins in developmental stages. In bacteria, DNA methylation can be influenced by various factors including growth condition, metabolic state, and regulatory mechanisms. So just to be clear now, DNA methylation, it is a general procedure that occurs in both prokaryotes and eukaryotes. But the only difference is that in prokaryotes we have DAM as well, which is DNA adenine methylation, where instead of cytosine, your methyl groups are attaching to adenine. So that's one thing that happens only in prokaryotes and not in eukaryotes. Eukaryotes strictly follow your cytosine methylation. Cytosine. It happens on cytosine. I'd also like to mention the origin of DNA methylation in bacteria. So, in 1953, the first evidence of DNA methylation was found in bacteria during phage infection. In this case, DNA methylation originated as a method to protect bacterial DNA from the DNA of the phage. The defense mechanism is termed as the restriction modification system, consisting of two components: DNA methyltransferase and restriction endonuclease. So, first, the methyltransferase methylates a specific section of DNA, while the restriction endonuclease cleaves the DNA based on the sequence. And as the bacteria consists of both the restriction and modification systems, they can effectively differentiate between the self and non self DNA to restrict the foreign or bacteriophage infection. So that's about it for DNA methylation. Thank you for watching.